So good afternoon, everyone, and welcome to your class in ADC 111, Prep and Animal Improvement. Um, last time we talked about plant breeding, and we still have some unfinished slides under plant breeding. So we are going to continue for this session, and I just thought that I have forget to actually talk take up the importance of biotechnology. So we're going to go back on that after plant breeding before proceeding animal breeding. And I will be making use of the time since we're running a bit of time already, and we are late by our timeline. As you notice, yung mga due dates ng inyong quizzes and final and midterm are actually due, no? Sa module 1, but we're not yet done. So I'll be fast on this. So I already also posted a video on what we have discussed last meeting, so please check it out. So in continuation on this, for this session, so we are now on the effect of reproductive behavior. So we have learned last meeting on the different ways of the monohybrid and the dihybrid crossing, the quantitative traits and the qualitative traits, no? And way the heterozygosity and homozygosity has been this one. So I have shown you this one, which is also talked about on your um, in detail sa previous genetic subject, no? Last time. So now to continue, what are these reproductive behaviors? So it's the behavior of the reproductive ability. So there are actually three ways to for reproduction of plants, and this includes self-pollinated, as we have discussed on the last video, cross-pollinated, and vegetative reproduction. So in the self-pollinated, it comes with the perfect flower. So last time we discussed the types of flower, whether they are male flower or female flower, imperfect or perfect. So for perfect flower with both male and female intact on its flower. Afternoon. So for this session, Micah, we're going to continue with the reproductive behavior. So I'm
Good
just,
afternoon.
yes, I will just going to continue with our previous discussion, and which is about plant breeding. So we have the pure line variety and the hybrid variety for the self-pollinated perfect flower. And in case wherein it is pollinated, like in the case of monoecious or monoaceae, dioaceae, and self-incompatible na mga crops like corn, no? These are done for the production of synthetic variety. So there are definitely heterogeneous population, not a pure line variety, unlike for the self-pollinated ones. And it's also a result of hybrid variety. It can result to hybrid variety, and if inbred development is also possible. So although the self-pollinated is a pure line variety, it will definitely produce hybrid variety also at the same time. So I hope you understand what it is meant, no? So I don't know sino itong pumasok. So if please write a name on your account so I will know who your name. So going back to the topics, no? We have the last way of reproduction is the vegetative. So no flowering or limited flowering. So this is definitely will result to clonal variety, or which means kung ano yung kanyang characteristic, that would definitely be its really characteristic. So, and it will also result to hybrid. So, in this figure, this is the pedigree breeding method, which is used in self-pollinated species to derive pure line varieties when it is desirable to practice selection in early generations. So, ito yung pedigree, which was mentioned on the first discussion last week, no? So, these are the cross-selected homozygous parents. So, the F1 for filial generation, and in on the second year, the F1 plants are grown, and these are all identicals as we have known from the crossing, the monohybrid crossing, na lahat ay identical talaga yung F1, and harvested sufficient if you, and then you harvest the seeds from this, and then you are going to again grow this into the second filial plants in wide spacing, and then select single plants for highly heritable traits. So magsiselect ka kahit ano yung basta nakikita mo na yung mga traits na gusto mo, then harvest seeds separately for each plant. So ito, harvest but separate, harvest but separate, and record identity and observation. So yung mga, ito ba mas matatangkad, mas madadaming bunga, and white colored or yellow colored ba, and so on. So for the next year, those ones selected are grown again for the F3 generation, no? So again, the same. Select desirable families and plants, mamimili ka naman, and then harvest them separately and observe for observations. And then on the year 5, i-grow mo na naman, no, and arrange rows to keep families together and identify good families, and select good plants from promising families, harvest individually and keep records. And then again and again, so pwedeng kahit ano lang piliin mo dito, di ba? So the next 5th, 6th, and filial generation, the bulk selected progeny are planted in row in F5 and F6, and then so yung F5 and F6 i-combine mo na, and then proceed to increase and test. Ito yung tinatawag na pedigree breeding. Now, the next is cultivar development for self-pollinated species using the pedigree method. So, from the germplasm, the cultivar mismo, local or exotic land races or wild relatives, this will produce hybridization, and parents are usually inbred. So, sa F1, in the nursery, all plants are heterozygous, and homogeneous population if parents were inbred. And on the second, F2, second filial generation, in the nursery, all plants are again heterozygous, and every single plant is different genotype. And then in the third filial generation, it's arranged in head rows, and you're going to select the best rows or the best plant within selected rows, then proceed again to the 4 head rows to the F4 or fourth filial generation. So this typical pedigree method selection in self-pollinated group. So self-pollinated naman siya, and each head row is called line. So mostly 6 to 7 filial lines are uniform enough for preliminary yield testing. So imagine it will take you 7 years for the traditional na breeding system. So ito, this one is the pedigree method. Ito naman for the bulk method ng self-pollinated species. So again, they are just the same, no? For from this part here um to the F1, no? Pagdating ng F1 F2, um you're going to collect equal amount of seed from each plant na since all plants are heterozygous. And in F3, instead of heteros, you're going to use the bulk population or repeat one or two more generation that follow heteros. So this bulk method for self-pollinated crop breedings has for the 6 and 7 F lines are uniform enough for preliminary yield testings, and therefore it's a less resource consuming. So now another one is for the single seeded descent, or we called as the SSD breeding method, used in self-pollinated species to derive pure line varieties when it's desirable to select from random homozygous lines in an advanced generation. So ito naman, the single seed descent. So how does it again, this is a cross-selected homozygous parents, no? So the F1 are all alike and should harvest sufficient. Again, you're going to harvest for the second year or the year 3, which for the second generation, and then you're going to plant them in high density and avoid selection of any type. Harvest only one seed per F. So lahat sila harvestin mo ng isang seed per F2 plant and discard remaining seed. So tig-iisa lang. And that's why single seed descent siya. And then grow one plant from each harvested seed, avoid selection as above, and then harvest one seed per plant again, repeat for 1 to several additional generations. So hanggang sa F4, F5th, and hanggang 6th, no, you grow space planted individual plants, usually from the 4th to 5th F filial seed, and select plants on highly heritable characteristics. So this time you're going to select na from 4th F filial generation and harvest seed from promising plants. So isa-select mo na. Then grow progeny rows. So grow them in rows. Roach off type plants, then select and box seed from progeny rows, then proceed to testing. So, so this is ito yung kaibahan ng SSD. So meron din tayong tinatawag na backcross breeding and recurrent selection, no? Wherein yung donor parent could have disease resistance, while the recurrent parent could have adopted but susceptible siya sa disease resistance. But ito yung ating ginagamit na parang variety, but then wala siyang disease resistance. So we need to, if you know, this is again a hybrid type or parang hybrid type na siya, and ito yung parent niya, no? So gusto mong ibalik doon kasi nawala yung resistance na, or it could be another also na breed, but then gusto mo silang i-cross para makuha niya yung disease resistance niya. So this is how it is done. So when you breed them, so ito supposedly is the R dominance, no, capital letter, and the recessive. So when you cross them, you got the F1. So supposedly meron na yung combination ng disease resistant na dominant and recessive na parent. So ang dominance na gusto nating kunin is 1/2 pa lang yung nakuha in the backcross breeding. And in the backcross first generation, again, you're going to get the recurrent parent, and then eto na naman ang kukunin mo. Iko-cross mo yung dalawa, ang result na nito is etong combination pa rin. In this part is 1/4 na yung kanyang donor parent. In this part, 1/8 hanggang maging 1/9 na as as you again cross this back cross 2 with the recurrent parent. And then back cross hanggang ilang beses, repeat back crosses, then self-pollinate again, test and multiply. So makukuha na yun niya ang, so let's just say high yielding siya. So makukuha na niya yung 1/2 hanggang 182. Depende anong cross ng kailangan ng disease resistance. So this is to transfer alleles, a small number of loci, from a donor pair into the genetic background of a reciprocal parent. Each generation of backcrossing reduces the proportion of alleles from the donor. So nagre-reduce siya to, and then from the donor D or donor parent by half, no, as shown here. So this is an example of a recurrent selection strategy with progeny testing. Many variations of this type of strategy had been devised also. So this is just cross-pollinating. Kasi kanina yung mga pedigree method, bulk method, and the pedigree method na self-pollinated yun. Ito naman ay saan na ba tayo? Sa cross-pollinating crops. So, in here pa lang, sa cross-pollinating crops, you're going to grow population and then intercross, allow random mating, so kahit saan lang dyan. And then, identify desirable plants. So, ayan, halimbawa, ito yung na-identify mo, yung naka-circle lahat. And harvest seed and retain records. And then on the year 2, you're again to grow, you are now to grow them in progeny rows. So, kung yung lahat ng letter C na galing nito, in-row mo siya. Yung letter A, D, E, F, B, for example. Then keep half of the seed in reserve and evaluate the progeny rows and identify desirable parents. So, pag napili mo na yung C, for example, at saka F, yung gusto mo na desirable traits, so yun na ang yung arrow row. You plant new cross block using reserve seed from plants that perform well in the progeny test and repeat. So this is for cross-pollination, which is different in the year 2 from the other self-pollinating crops na pedigree and bulk method. So this is just a schematic simplification of the development ng synthetic plant variety in an outcrossing species. So the scene 1 generation is produced by random mating of reproducible components or the inbred lines or clones. And if found to be desirable as a new plant variety, it can be produced and sold by repeating the identical crossing block. And this type of breeding method is most practical in perennial forage species. If adequate seed cannot be produced in scene 1 generation, the scene 2 generation harvested from scene 1 generation may be used instead. So after nag-random mating siya, no, then you're going to select the best, no, from here, and then against mag-reserve ka, and then ito na, itetest mo na naman hanggang napili mo na yung kanyang, or you can even repeat identical steps to produce. So again, in another simplified development of hybrid plant varieties, for instance, in corn, the parents are A, B, and AC, and then after that, you're going to match them, develop inbred or clonal plants through mass or recurrent selection, and maintain the inbred or clones, then grow in isolate pairs, so naka-isolate pair sila, or emasculate female plant, and harvest seed from female plants. So, ito nang dalawa naman, you're going to grow hybrid test and evaluate yield and other traits and repeat over multiple years and environments to identify the best synthetic varieties. And again, repeat them. So ito, A and B na, repeat hybrid cross on much larger scale to produce for seed for commercial production. So yung type of seeds actually ay we have even the for the types of seed, if you have learned in your previous lesson, meron tayong tinatawag na um, foundation seeds or breeder seeds. Okay, hanggang sa available na yan sa market. So again, in other crops, the parents may also be clonally propagated. Parents are grown in adjacent rows for crossing, and the female parent is emasculated so that it will not self-pollinated. So last video we have learned what does self-pollination means, and for seed harvested from the female parent is tested in performance trials also. And if the hybrid variety is successful, then the cross is again repeated on a larger scale for commercialization. So with traditional backcross breeding, this actually happens, no? For the from backcross one to again with the recurrent parent F1. So yung kanyang backcross na yung nais mo na desire, and from 50 magiging 75, 87, 93 to hanggang 99% in the year 7. So it will really take years when you actually breed a plant. But using technology like the molecular markers, no, this allow visualization of genotype. So nakikita mo na dito magkalay ng mga genetic or genotypes na characteristic. So for example, ito lahat sa first line, dito siya kasi may mga bandwidth ito. So ang magka-line, yun lahat ay mga recessive na R. At ang magka-line dito sa taas ay mga dominant na R or capital R. At ang merong ganito, dito na line, R actually na meron na siyang gen type na both dominant at recessive na R. So if ito yung target mo, alam mo na dito na siya na marker. So this is a gel electrophoresis of DNA markers. So with using this, we can now see genotypes to accelerate the breeding cycle instead of crossing them again and again from first filial generation to the nth level or to the 7th year. So again, this is another visualization of SNP markers on chromosome 1 for a set of soybean varieties. So ang daming linya, no? So each column represent a locus position on the chromosome, and each row represent a different soybean variety. So imagine, first row, second row, iba-ibang variety yan. And the predicted value of each allele is determined by testing a reference population where phenotypes are known, and then the predicted genotype value of each soybean variety is then derived as a summation of predicted allele values and varieties with the highest overall genotype values are selected. So in computerized generation na molecular marker, you can also predict kung anong if you're going to combine these genotypes, then the products can be predicted. So there are also other ways of breeding methods, which we will not discuss further, but we have talked about apomixis and mutation breeding, vegetative reproduction. So to summarize, plant breeding mostly uses sexual process to recombine genes and alleles and crops, and the goal is to usually cultivar with improved traits, and self-feeding crops are improved using the pedigree method or the bulk method, such as the single seed descent. So recurrent selection and backcrossing is also useful tool, especially for outcrossing crops. Synthetic varieties and hybrids are the product ng breeding outcrossing species. While mutation or vegetative cloning and apomixis, so I hope you still remember what is apomixis in our previous discussion. It's also used in breeding. And marker assisted selection can speed up breeding cycles. So I guess that's all for this the continuation of plant breeding. So if you have questions, you may ask your questions now.